and he received his bachelor and master exam from Manchester University, Great Britain, and his PhD from Justice Liebig University in Gießen, Germany, and his second doctoral degree from Zurich University in Switzerland. And Nigel Crompton, he teaches biology, genetics, molecular cell biology, and bioethics, and questions of origin and neuroscience at Cornerstone University. And he has published 100 scientific papers, half of which in papers with peer-reviewed systems. And Nigel has also developed a biomedical test for predicting how patients respond to cancer therapy. And Nigel Crompton, he has also for many years been associated with the German creationary organization Wort und Wissen. And today, and since long, Nigel has been doing lots of work, interesting work, and written many papers on the Mendelian mechanisms and how they also lead to speciation. And that will be the title of this talk, Mendelian speciation and the origin of species. So, welcome, Nigel.
Thank you so much. Thank you for the invitation to come here to share this information with you. I want to talk a little bit about Mendelian speciation and the origin of species. Ola, thank you so much for saying all those nice things about me. I was in cancer research and radiation oncology for about 20 years, and I've been a little over 20 years, and I've been teaching now for a little over 20 years. Is everything fine? Can you not hear me? Is this loud enough? Yeah. Is this better? Good, thank you. This is the frontispiece of Darwin's famous book, The Origin of Species. It actually has a very complicated title, very long title. It's On the Origin of Species by Means of Natural Selection, or the Preservation of Favoured Races in the Struggle for Life. And if we look at that, so there's a double title in one sense, but before the or, he makes this statement on the origin of species by means of natural selection. What Darwin is saying is that natural selection gives rise to species. That's his whole premise. You know? Many folks get very confused about what natural selection is. Due to the notable esteem granted to Darwin's ideas, they seem to imagine it's an enchanted process, which can cause new species to magically appear. If you hear many nature documentaries, you'll hear the announcer sort of going through and talking about this whale or that bird or whatever, and then suddenly say, and then it arose through natural selection. But this isn't right. You know? Darwin would be, of course, gratified to know he has such ardent fans. That's why there's a lot of thank yous there for all of his fans. Natural selection is simply a form of selection. It selects things which are favoured. It is not magical, and it can select nothing that doesn't already exist. If differences arise in species, it can select fittest individuals, but selection cannot produce those differences. It's unable to do that. There are other forms of selection: sexual selection, artificial selection. And those are also completely incapable of giving rise to new species. Here too, it is just as true. Only if differences are present can the fittest of already existing alternatives be selected. So what then is the origin of species? The goal of this talk is to examine the origin of species, but based on field observations. Now, we would like to emphasize that because hypothetical mechanisms about speciation based on speculative roles and mutations, they overwhelm our biology, genetics, and evolution textbooks. They're all hypothetical mechanisms. Yeah? Trying to account for novel species by mutating or altering existing species' various genes is somewhat like trying to produce new novels. by mutating or altering existing novels various words. It's not really a very promising approach. Mendel's studies were conducted standing on the shoulder of giants. It's quite a famous statement that. He readily acknowledged his debt to various well-known botanists, including Gärtner and Kohlreuter and others. These early pioneers had devoted their careers to studying hybrids, new cross plants. Many of them had reported observing the hybrids tended to revert in appearance to their parental forms. And I'll show you a number of examples. This reversion phenomenon was so common that Mendel referred to it in his paper as das Entwicklungsgesetz, the law of development. Yeah, of course, by that he was referring to the temporal development of populations. He wasn't talking about embryonic development. The law resulted from systematic genetic changes which occurred in hybrid populations over a number of generations. And Mendel understood, I'll talk a little bit about this later too, he understood it was caused by loss of heterozygosity. Now when we talk about loss of heterozygosity, we mean in the same breath, without saying it, gain of homozygosity. It's not as though we don't have anything then. The heterozygosity is replaced by homozygosity. So what is speciation? Do we mean to mutate a trait? Do we mean to mutate a character? I'm not so sure, but maybe I should just mention this because different textbooks have different definitions for these things. What I mean by a character is actually something rather ephemeral. Yeah, it's like hair colour. Hair colour is, well, we all know what hair colour is, but it's all sorts of different things, isn't it? It's red or black or blonde or whatever. But so we have hair colour, that's the character. What we actually see, the red or the black or the brown, that is a trait. And there's a difference between them. And it's very important to understand that difference, to understand what's going on here. Yeah. So when we mean by speciation to change a character's trait, so instead of having a red-haired person, we have a black-haired person. Is that what we mean? Do we mean to actually mutate a character? Well, that's a very interesting concept, so that someone maybe doesn't have hair colour at all. Maybe they're bald. Yeah. Is that what we mean by a species? A new species? Do we mean to maybe mutate many traits in different characters, all within a short time? Perhaps that's a little closer, perhaps. Yeah. Are species simply accumulations of mutations? Why would I say that? Because the statement repeatedly heard is that speciation is caused by mutations, DNA sequence changes. In one sense, it could be true. If I want to change a car repair manual into one of Shakespeare's plays, or a biology textbook, or a theological exposition, all I have to do is to change enough letters, and also the number of letters, and eventually I'll meet with some limited success. Right? But it's not really how speciation occurs in nature. Here are some examples of gene change, and some extraordinary ones. We got the bottom here, on the bottom left, piebaldism, and then this young lady, she has heterochromia. Her two eye colours are different. There's the werewolf, the hypertrichosis. And these are perhaps somewhat extraordinary mutations, but they're all clearly human. There's no question about that whatsoever. We've not caused a new mutation, a new species by this mutation event. Detrimental mutation events can make sick. They're still 100% human. You should never doubt that. In fact, you can take Drosophila and you can do all sorts of exotic things to it and try to mutate it to death, and it'll still stay Drosophila melanogaster. Mutate as much as you will, you always get the same species. Similarly, alter the rate of gene expression as much as you will, and you always get the same species. Mutations, they tend to degrade genetic information. Now this is an important point, which I would like to point out. Epigenetic modifications or transpositions, yeah, they can be programmed to alter traits, yeah, but it doesn't actually give us a new species. Do you remember our famous peppered moth? And we had the classic sort of black-white morph, and then the black, it's still the same species. Now, epigenetic modifications or transpositions can result in the expression of latent biogenetic potential. They're very important. Yeah, this can significantly contribute to novel phenotypic change and speciation, but these are players in a larger scheme. And I do not want to question in any sense the importance of these players. They're very important players, but they are players in a larger scheme. So if it's not mutations, if it's not simple trait changes, then what is it? Well, two practical questions need to be addressed. I think it's only fair when we're thinking about speciation, because a lot of it, and particularly in textbooks, it's all wiffly-waffly, speculation. Let's get down to the brass tacks, shall we? Can we repeatedly make a species? Is that possible? Yeah, and then what genetic mechanisms were involved? These first examples are in plants. These are actually primarily artificial examples, but it goes on a lot in plants. I'll also give you some animal examples later. But you see the grapefruit is one species. It's actually a hybrid of two others. The same with the cherry. The same with the banana. The same with peanuts. Yeah. Let's move across to the animals, shall we? Here's an old world tufted duck and an old world pockard. And the hybrid, you can see, the Ithia hybrid. And there's a new world lesser scalp, looks exactly like it. All we have to do is we just simply cross these. Hybridization reveals already existing species. Another example here, it's another one from ducks. I'm grateful to Siegfried Scherer for these duck pictures. The shoveler and the cinnamon teal, and you can see the hybrid at the bottom, and you'll notice the hybrid looks exactly like the Australian shoveler, which actually lives somewhere quite different. And what I find very fascinating about that particular picture is you can see the white bar on its face. It wasn't present in either of the parents. It was hidden, and now it suddenly appeared. I'll talk a lot more about that. In some science papers recently, there's been a lot of discussion about whether you can get these hybrid species. You can see the green bird in the middle. That's the golden-crowned mannequin, yeah? And on either side are two species which, when they're hybridized, give rise to that third species. Another example, another bird example, even a lot of bird examples. Here's a couple of warblers at the top, the blue-winged and the golden-winged, and when they cross, they produce Brewster's warbler. And when that crosses with the chestnut-sided warbler, this also came out just recently, you get them Burkitt's warbler. And what we're seeing here is species can arise via hybridization. Yeah? There was another paper which came very excited when this one came out. It's from about the grey snub-nosed monkey, and it turns out that it's a hybrid of the golden snub-nosed monkey and the black snub-nosed monkeys, and they give rise then, when they hybridize, to the grey snub-nosed monkeys. These are real tangible examples of species. We're not talking about some wishful thinking here. For many biologists, hybrid species are actually something of a paradox. Why would that be the case? Because prominent leaders in the field, like Dobzhansky and Meyer, very famous biologists, for those who are not necessarily familiar with population genetics and so on, they have persuasively argued for decades that species are reproductively isolated. If they do produce offspring, then the hybrids are sterile. Well, it turns out to be true, but only about half the time. Hybrid species are turning up everywhere. And very tellingly, hybrid species also turn up where speciation abounds. So what's that all about? I think we touched a little bit on adaptive radiations. I'll give you a few examples to tell you about that. Speciation abounds where in certain circumstances where suddenly there's an explosion of new species. Very extraordinary. But how can, clearly when we see these species there's a lot of differences. How can many genes change all at once? Well, this of course is possible at meiosis. During pachytene of prophase 1 of meiosis, all the homologous chromosomes pair up and they form bivalents or tetrads, depending on what you want to refer to the chromosomes or the chromatids involved. They're carefully aligned using something called the synaptonemal complex, which in and of itself would be a wonderful example, not quite as fabulous as photosynthetic apparatus, but this particular apparatus, it lines up those hundreds of millions of base pairs to the very base pair. And it has to do it technically every time, absolutely exactly, and yet it does it. Very, very fascinating. Carefully aligned using synaptonemal complex, crossing over the exchange of non-sister chromatin arms occurs, the exact complementary base pair. Recombination of genes does not involve just one or two genes, but the whole genome. Speciation involves many traits simultaneously changing. And this is the way it can happen. We've heard a little bit about hybrids. Here's a couple of fun hybrids. We've got a picture of a Hebra at the top left, and a Zorse, the bottom right, and of course they are the hybrids of the horse and the zebra. These funny names they get. The male parent is included first, and then the female parent afterwards. So the Hebra is a cross between a male horse and a female zebra, and the Zorse is a cross between a male zebra and a female horse. What is interesting is that with these crosses, if you actually compare the hybrids, they look a little bit like the male parent, a little bit more like the male parent. If you look carefully, you'll see that. The very famous one between the lions and the tiger. The liger is very famous. Obviously, that's the male lion and the female tiger. What is also interesting about the liger, I just mentioned this, is they are absolutely massive. They lose growth control and they become huge animals, very, very spectacular. Not so with the tigons. That would be the male tiger and the female lion. And just so the birds don't feel left out, we have crosses here between swans and geese. Yeah, the swoos and the guan. I'll leave you to work out who the male parent was and who the female parent was. Here's a nice example in birds of paradise. I'll mention a little bit more about them later during the talk as well. So we have the king bird of paradise, and it crosses with the magnificent bird of paradise, and it will give two hybrids. And obviously, if you look at these carefully, you can see perhaps easiest by the amount of curling on the tail feathers. The one looks a little bit more like the king of paradise, and the other looks a little bit more like the magnificent bird of paradise, and you can work out then where those two crosses come from. And as far as flowers go, these are the tarweeds. You find them on the coast of California. And a lovely bird, the golden plover, it flew them across to Hawaii. So this is one parent. This is Anisocarpus cabridus. This is a flower you always want to use if you want to find out if that someone special in your life who you're wondering about likes you or not, because it has one ray floret. She loves me. Here's another Tarweed, this is Colchicum murii, named after a very famous, actually two famous botanists. No ray florets, but somehow or other, we're not quite so sure if it happened in California or if the seeds of both were carried across to Hawaii and they crossed there, but it gave rise to a fabulous group of plants in Hawaii, the Silver Sword Alliance. Yeah. So these are the chromosomes of the cross when it's made. That's the cross that started it all. And what you're seeing here is referred to as a hybridization matrix. And it shows abbreviated the various different species of plant and other plants with which they can hybridize. Now remember what they came from. That one-rayed and that non-rayed little sort of composity there. Unfortunately it's not in the next picture, but I'll show you then a picture of what some of these species look like. The species, they could all form hybrids. Well, almost all. There's one which is extinct, so you can't say much about that one anyway. These plants look incredibly different. We're talking about trees and small bushes and cushion plants and vine-like plants, lianas, and the silver sword itself is an extraordinary plant. Very, very fascinating. You can see sometimes they have 14 chromosomes, and interestingly enough, sometimes they have 13 chromosomes, and that doesn't seem to bother them at all. They can still make hybrids. Thank you very much. Now, what do these hybrids tell us? Well, a species is a set of characters. 2 copies of each character are present, one from each parent. Okay. Characters can have alternative traits. I think that was something we already knew. Usually, only one trait at a time is expressed. And if alternative traits are present, one is dominant. Now, this is quite important. Novel combinations of expressed traits produce novel phenotypes. The information is there. When I showed you the Zorse and the Hebra, all of the information in those 2 animals for horses and for zebras were present in the hybrid. And if you selected them, you could actually go back to a horse, or you could go back to a zebra. All the information was there. It's telling us that all the information can be there in the genome. Yeah? And if we recombine it, we're going to get new species. The information for speciation is already present in the genome. Nothing new needs to be made. No mutations have to be, contribute. So where is speciation seen? I've mentioned these, the adaptive radiations. I'll just very briefly take you through a couple. The most spectacular examples of evolution are the adaptive radiations, which include actually a number on Hawaii, the plant alliances in Hawaii, the silver swords, which we've already met. I'll show you some pictures of those, but others too. The picture-winged Drosophilas, they're fruit flies on Hawaii. And fruit fly species on Hawaii are so numerous, and Hawaii is really not very big. It's almost as though in every tree you have a new species. It's not quite as bad as that, but almost as bad as that. There are a huge number of species. The cichlid fish in the East African Rift Valley lakes, they're very famous. For those of you who don't know, I'll show you a couple of pictures. The dewlap lizards of the Greater Antilles, finches in the Galapagos, they're Darwin's finches, and they're very famous adaptive radiation. But there's a much more delightful, spectacular radiation of finches, technically of true finches, because Darwin's finches are actually tanagers, but whatever. Yeah. But a lovely radiation of finches in Hawaii with which include the iiwi, the honey creepers. It's the iconic finch of Hawaii. So these are the silver swords, and at the top left you can see the Wilkexias, quite extraordinary. This one is branched, but very often it's just a single stem with these leaves coming out. I suppose it looks a little bit like almost like a palm tree or something like that. And then when it flowers, it sends up this rocket, this panicle of beautiful, beautiful flowers. The silver sword, you can actually see it's on the top right, and you can see at the bottom those leaves, they have lots of little hairs on, so they reflect the sunlight. And the sort of narrow, like a sword, that's where it gets its name from, silver sword. And usually that's all you see. And you can see quite a few of them. They somehow or they eke out an existence on the lava, not the hot lava, obviously. And they'll grow there. Very fascinating to see them. And they're monocarpic, so once in their life they flower. And when they flower, they send up this panicle here with these purple flowers on. You can see in the inset next to it those purple flowers. To give you some idea of the size, if a person was standing next to it, they'd be about the same size. So these are very big flowers, plants. But you can also see, I mean, if you look at these two corners, enormous differences. Down here, the bottom right, you've got a tree, and then you've got these bushes, and in the center top, you've got this the vine type, the liana type, and then a little cushion on all of these different flowers. And they can all cross with each other. In fact, if you go to, you have opportunity and the pleasure to go to Hawaii, if you go to Maui, to the national forest there, Haleakala it's called, on the top of the volcano. If you go to the ranger station, they literally grow this flower, the silver sword itself, and then something which looks rather like the one at the bottom center. And they grow them there, and they allow them to hybridize, so you can see the hybrids. This happens quite naturally. So there's a sort of a mini hybridization matrix shown down there. It's not the full picture. There's far more to it. And here are different Drosophilas, different shapes. The classic one used in the laboratory is somewhat smaller. You get all these different species. They're called picture wing Drosophilids because they have these patterns on their wings, which are meant to sort of be little pictures, and they change from one species to another. The East African Rift Valley lakes, they're sometimes referred to as the African Great Lakes, have these fish swarms in them. I think, Peter, didn't you mention them today? Yes. Yeah. And if you look at them, you can see on the left side there's a species from Lake Tanganyika, and on the right side species from Lake Malawi. And you'll notice that they all look rather similar. However, genetically, all of the species from Lake Malawi are more closely related to each other genetically than they are to any of the species in Lake Tanganyika. And I think that's what it's showing us, is the information is already present in the genome. It simply has to be revealed. Similar kind of thing, a little more complicated, this one. This is the dewlap lizards. When they try to attract their mates, the males, they have the throat fold of skin which pops out in bright red or orange. And then apparently all the ladies, it makes them go weak at the knees. Something like that anyway, but it's the way they try to run their courtship. And you can see, I think, perhaps the hardest one to see is the trunk ecomorph at the top right. Obviously the trunk crown ecomorph, that's green, which ties in nicely with the colour of the plant, and the blue there is meant to make it look like a flower, so that flies will come along and that they provide then sort of meals on wheels or something, lunchtime. Yeah? But what is interesting is these different ecomorphs, they all look quite different. They all arose on not just one of the major Caribbean islands, that would be Cuba and Hispaniola and I forget the other two, Jamaica and what's the other one? Puerto Rico. Yeah. But those 4 islands, quite independently of each other, on all 4 islands, these different ecomorphs arise. The genetic information is present and comes out, and when the species arrived at these islands, they then radiated out. They produced lots of different ecomorph species. These are Darwin's finches, quite famous for those. Or the finches are quite famous for Darwin, or one way or the other, on the Galapagos. And although they're interesting, it's an interesting example. If you go to Hawaii, you find another finch radiation, but I think far more spectacular, with fabulous colours and very prominent beak bills and all sorts, even bits of, not fluff, but sort of a feather crest and stuff like that. These are adaptive radiations. This is speciation happening, and happening quickly. And these, for instance, the silver swords, they can all hybridize with each other. When you do the genetics, the genomics, what do you find? Adaptive radiations are consistently associated with minimal genetic change. What is surprising is there's insufficient differences in the molecular sequences to clearly separate member species, because they're so similar to each other. Adaptive radiations demonstrate evidence of inherent genetic subroutines. For instance, the ecomorphs of the dewlap lizards, or those cichlid fish, you kept seeing the same thing coming out. The information is there already. Nothing has to be mutated into existence. It's already present. So how do adaptive radiations take place? How can we get major phenotypic change, but with only minor genetic change? Gregor Mendel, the father of genetics, provided us with a simple, clear answer. He demonstrated that because genes have at least 2 alleles, enormous phenotypic variation is available for profuse speciation. So here's Mendel, and he wasn't a monk, he was a friar. Very important difference. Friars, they still took a vow of celibacy, but they got involved in the community and tried to help folks rather than retreating into a monastery. And in his monastery garden, or technically abbey garden, he crossed then his peas, his garden peas, and you can see he has purple ones, and he had purple flowering peas and white flowering peas. And when he crossed these, all the progeny were purple. There weren't any white progeny. Now, in one sense, that's very sad. We should all have a little funeral for the white flowers. But when he self-crossed those purple children, out popped some white flowers. The ratio of 3 to 1. He actually did this for 7 different characters. And what you're seeing when you see pictures of these characters is on the left side, you always see the dominant one. And on the to the right of that, then you see the recessive traits. And he did it for all of these, and he found consistently the same thing, that after his first generation, one of the traits disappeared. He called that the recessive trait. The one that was always shown, called the dominant. I'm sure many of you are very familiar with that. When he self-crossed the children, now, probably worth just mentioning that the parents were what is called pure breeding. So if it was purple, it would only ever get, the parents would only ever give rise to purple kids. And the white flowered peas would only ever give rise to white. Well, technically they only could ever give rise to white. But when he crossed them, then there was a mixture. Now, they're either pure breeding, or if they're a mixture, they're called hybrids. And that's what we see, that's the phenotype. The genotype is also given a name. If they're pure breeding, then they're homozygous. But if they're hybrids, then they're heterozygous. And so we can see, if you take the children from that first cross, they would have all have been hybrids, all heterozygous. If you self-cross the children, then what you got was this 3 to 1 ratio. And What's important just to point out here, so you understand what's going on with speciation and how it works in nature, is we looked at one gene. What we could get are two phenotypes, in this case purple and white. So what do you get if you do it with two genes? It's a little more complicated in this particular case with two genes, two different characters. In this case it's seed shape and seed colour. But if you look at what is referred to as the Punnett square at the right, you look along what's called the leading diagonal, you can see there's yellow and round and green and round and yellow and wrinkled and green and wrinkled. Two genes give us four phenotypes. And what happens if you do the three gene situation? I've just labeled them A, B and C to make life easier. If you look down the leading diagonal, you'll find that there are all the different possible phenotypes, different types of phenotypes. There's 8 of them. So we went from one gene which had two genes which had 4, 3 genes which have 8. Do you see what's happening, this exponential increase? Yeah. Mendel's credited for 2 laws, which he never actually stated. He did mention 2 laws in his paper. The first one I mentioned previously, that's the law of development, Entwicklungsgesetz. And the other one was, he said it 3 times directly in his paper. He called it a law. He's never recognized it for it. I just call it in English the law of exponential trait combinations. And basically what that law says is as you increase the number of genes linearly, you increase the different combinations, the alternative phenotypes, you increase them exponentially. So if you have just 9 characters or genes, that would give you 512 phenotypes. There are very few families which have 512 different species in, and that only requires 9 genes. 20 genes, 20 characters, you can get 1 million different phenotypes. There are no families on Earth which have 1 million different species in. There are some which get up to the thousands and rather unique ones. And all that you require to do that is 20 genes with alternative alleles. Surely then an enormous amount of DNA is needed to account for all the observed biodiversity? Not at all. This means of creating enormous amounts of biological variation is absolutely brilliant. We wouldn't expect less. All is required for the vast numbers of species in all of their variation and uniqueness is simply 2 alleles at each gene. Now there are more alleles and lots of genes, so obviously there's an enormous amount of potential here. Populations have far more than 2 alleles at most genes, so there's the potential for exceedingly more we could ever ask for or imagine. A reasonable question to ask at this stage is, can major genetic potential be hidden in the genome? You remember those tar weeds we saw? Those little sort of dandelion type things on colour, and what they became in Hawaii, is that even imaginable? So let's try and tackle that question next. At the trait level, we know that's true. We've just discussed it. Dominant alleles hide recessive alleles. Definitely genetic potential can be hidden. It's also true at the character level. If I have any genetic enthusiasts here, they'll all get excited to hear about epistasis. I don't want to say too much about it, maybe just mention it very briefly. At the cell level, we know it is. We all know it is very well because we all start off. Now you're going to have to remember back to your youth, back to your very early youth, when you were a zygote. Do you remember you were just one cell type? And then there were actually at least 200 different cell types hidden in the genetic information of that one cell type. So yes, we can hide genetic information. At the organism level, it's also true. Gender and metamorphoses are also hidden. I won't go into those details because I just want to show you how some of this works out. So here we have the wild mustard, Brassica oleracea. And if you select it, you can make broccoli, cauliflower, collard greens, Brussels sprouts, kohlrabi, carrot and cabbage all out of wild mustard. The information is there, it's just waiting to be expressed. And I'm very grateful for Peter, for I think he also showed us this example. So we know that the grey wolf is the ancestor of all our dogs, but they show so many different types of breeds. Of course, that's only a picture. Does it really ever happen? Well, let's check out a cross between a poodle and a wolf. The poodles here, they're the two black dogs, and the wolf is that, what's the name of it, that particular colour? Oh, well, it slipped my mind now, but this is the wild colour of, agouti is the name, yeah, the agouti colour of the wolf. And if you do the cross, that will give you that first generation where you lose everything, right? Do you remember when Mendel lost his white flowers? So what we get in the first generation is this. Oh, they're very cute. That's what we get. But in the next generation, we're going to get more, aren't we? So let's cross them again. There's experiments which were actually done. I forget where they were done, somewhere in Germany. Out pops all of this. The information is there, it just needs to be brought out. Can major phenotypic potential be hidden? Well, at the character level, I think I've already mentioned that, these epistatic genes. As this is rather complex and as you had to wait so long, I shouldn't mention too much about this, but there are a couple of important things I do want to mention. One of them is that typically with dominance and recessive, you will see all of these new species very quickly. Under certain circumstances, but typically you'd expect to see them quite quickly. So why do we sometimes not see them for a while and then see them later? So let's just discuss that briefly, because I think that's got a lot to do with Peter's transposons and stuff like that. Dominant epistasis can reveal the repressed traits, but that's typically only within a few, or even just one generation. Recessive epistasis, however, can hide different traits for generations, hundreds of generations. Here's one example with Labrador dogs. Yeah, so we know we can have black, we can have chocolate, and we can have golden Labradors or retrievers, one or the other. But if you just take those golden Labradors and breed them with each other, you will hide the black and the brown. You'll never see it in the progeny. It's there, but it's hidden. And you will only see it later if under certain conditions and you do a correct cross, you will then be able to bring out those hidden traits. They can remain hidden, as I say, for hundreds of generations, and they need to be activated. That's just basically saying that, because I want to bring us back to our two tarweeds that I showed you earlier on. Remember that tarweed could make trees. The information for the tarweed is actually in that tree. Can small plants like tarweeds give rise to big woody plants like silver sword trees? Can small plants like tarweeds carry, concealed in their genome, significantly different phenotypic information? Well, the woodiness, specifically of silver swords, hasn't been investigated yet. As far as I know, it may actually be under investigation at the moment. But it has been investigated in botanists' favourite laboratory plant. It's called Arabidopsis thale-cress. It's a little weedy thing, and you find it probably growing here in Sweden. Certainly we have it as probably an invasive species in the US. But Arabidopsis, very small plant, and what you have to do is you have to inactivate. So So you could switch them off using transposons, or you could even mutate them. But if you inactivated those two, yeah, then suddenly you get a woody plant like an azalea. So on the bottom left, you can see the Arabidopsis. This is the wild type plant. This is the classic Arabidopsis. And you can see it's quite small. In fact, it's so small you can grow quite a lot in a petri dish, which is why it's rather liked by botanists to do molecular genetic studies and things. In the middle is what happens to that plant if you inactivate those two inhibitor genes. You get this thing looks like an azalea. If you look right at the very top, you can just about still see it actually is Arabidopsis. And then on the bottom right, you can see the wood, which wasn't there and now suddenly is there. Exuberant iron radiations, especially in plants, they're abundantly documented in the literature. Micronesia has hosted many. Spectacular adaptive radiations of Sonchus and Echium have occurred, and these also display the unexpected appearance of woody phenotypes. Repeat appearance of the woody phenotype, it suggests there are selectable genetic subroutines. The information is already there. It was laid into the genome right from the get-go. Yeah? We don't have to mutate things into existence, which is such a bizarre thought. It seems highly unlikely. So this is the prickly sow thistle on the left, and you can see it looks a little bit like a dandelion, but it's got thicker, so slightly more prickly leaves. And if you let that one go to the Canary Islands and get there established, then all of a sudden it's going to turn into the Canary Island tree. And if you look at the flowers at the top, you can just about make out all those dandelions at the top. That's the woodiness which came out. Here are, on the top left, is Viper's buglos. The Natterkopf is the German name for it. It's a beautiful wildflower. It's my favourite wildflower. It's gorgeous. It forms these big panicles with cymes coming out of flowers which are blue and pink. It's a very beautiful flower. But when Echium arrived at these islands here, suddenly you get it's like the third along from the top, those blue things, that's called the Pride of Madeira. And they are really proud of this plant, as it looks spectacular. Look down the bottom left here, look at these things. Pop out. All of that information is sat in the genome waiting to be expressed. It's not normally, it's just waiting to be triggered. Yeah? Woody members of the genera Sonchus and Echium, native to Micronesia, have arisen from herbaceous continental progenitors. Convergent de novo multiple mutations seems a rather unlikely explanation for this change, because it's occurred more than 1,000 times on 375 islands, not in the same species, in all sorts of different species. Insular woodiness appears as a result of activating expression of genes already present in herbaceous progenitors. And that's the statements of the botanist studying this. So can major phenotypic potential be hidden? It can certainly be hidden at the trait level, at the character level, at the cell level, at the organism level it can be. Here's one fascinating example. You may be familiar with this. This is the Mexican water monster. Yeah, the axolotl. It's the one on the bottom left. It's a neotenic salamander. That means that it actually reaches sexual maturity before it turns into the adult. And so it never turns into the adult. Well, when they worked out what was happening physiologically, they realized that what caused this was a lack of thyroid stimulating hormone. So somebody came up with the bright idea, right, let's give these axolotls some thyroid stimulating hormone and see what happens. And that black animal there is an animal which technically doesn't exist. And it shows you that the information for these creatures, it's sat in the genome simply waiting to be expressed. And under the right conditions, if it is, it can all start to pour out. Species give rise to families. Hybridization memories, they confirm life is a forest of families. I think people like the idea of it being a forest of bushes or something, lots of bushes of life rather than a single tree of life. Yeah. And this was the silver sword hybrid matrix, just as a reminder. Here's another example, and the reason I'm showing you so many examples is to try to emphasize the fact that this is the norm. This is how speciation normally happens. It's not something exotic. Yeah. So here's a bunch of marine snails. The whole matrix actually has 60 species in, but in the meantime 5 have gone extinct. So there's only 55 species now left in this family. They're all so similar, and they all have this, can you make it out? There's a sort of a row of holes. They're technically called tremata. There's an arc of tremata in these. The inside of the shell is a beautiful mother of pearl. It's a very unique mother of pearl because it's not just pearl coloured, but it also has iridescence. It's due to the way the structure was made. That's another pointer to our Lord's fabulous design, because everyone would like to mimic it because it's both beautiful and very, very strong. But these are called the abalones, and you can see that lots of them can cross with each other. In fact, they're technically so similar. They're definitely different, but they're so similar they are placed all in the same genus. And there's nothing like it. There's them, and that's it. There's nothing which sort of has half a row of tremata or something like that, which might have given rise to them. They're a unique family, all on their own, a Baramin or Genesis kind. And they kind of poo-poo the idea that hybrids are sterile. The abalone you see here is a 4-species. It is a lab hybrid, but if you take a red abalone and cross it with a white abalone, and you take a pink abalone and cross it with a flat abalone, and you cross the 2 progeny, and you get the 4-species hybrid. They're obviously fully fertile. Now there is a very, very famous natural history promoter from England. Oh, incredibly wealthy family. Anyway, they sent Ernst Mayr off to Papua New Guinea to check out the birds of paradise there, because there were some extremely rare species, and they wanted to be able to find them because you could sell the feathers of these extremely rare species at an absolutely ridiculous price. The feathers were used to adorn ladies' hats, millinery. And obviously, if you were wealthier, then you had fancier feathers in your hats. It was kind of a fad. It almost caused the extinction of the birds of paradise for a while. Really kind of sad. There's lots and lots one could say about the birds of paradise. But he went to look for them and he couldn't find any. And he suddenly realized what's going on. Oh, these are hybrids. So this is Ernst Mayr, despite his reluctance of hybrids, creating here a hybridization matrix. And you are not expected to read this next slide, thank goodness, but it has listed all of the different hybrids. At the time they were thought to be separate species, so they given a species name, but technically you never give a hybrid a species name, whatever. This is then one way of considering the hybrids and the hybridization matrix. And you've got all 40 species of birds of paradise here, and whenever there is a cross between separate genera, I've highlighted that with a black line. And you can see there's I think 24 of them. But what I want to do is this is going to require thinking caps and stuff like that, but I want you to think about what this might be telling us. So I'm going to recognize 2 kinds of hybrids. I'm going to recognize hybrids between close sister species, and for want of a better term, I'm going to call them proximal hybrids. And then I'm going to recognize hybrids between distant species, and I'm going to call those from different genera or whatever, or even different species complexes, whatever, as shown here. Yeah, I'm going to call those distal hybrids. Now, if these arose via mutation, these hybrids, they're very different. I'll show you a couple of examples. What you would expect to see is an accumulation of lots and lots of differences. So the hybrids should show, woo, there should be a bit of a waterfall of an enormous species. Yeah. Or, if they arose via this Mendelian method, yeah, then they should start to become more dominant, pan-dominant homozygous, kind of go back to the original state. So what do we see when we actually look at these? Now here's one example of proximal hybrids, and you can see Ragiana bird of paradise and the blue bird of paradise. If you ever want to spend a fun sort of half hour, you can go to YouTube and look at the birds of paradise and all the fancy ways that they caught their partners. It's extraordinary. They build little sort of ballerina type stages to dance on and all sorts of stuff. But this blue bird, it hangs upside down with its feathers all displayed, and it hums. You can actually hear this on the YouTube video. And it's a very beautiful bird. It's remarkable to see it when it's displaying like that. But when these two cross, the Ragiana bird of paradise, that's the one which is on the Papua New Guinea flag. That's their national bird. And when the two cross, you get Captain Blood's bird of paradise, and it sort of looks, because of its colour, this is halfway between, right? This is a proximal hybrid because same genus. And that's just to say about different the proximal hybrids and what one might expect. For instance, in red, one would expect significant differences if species arise via multiple mutations, not significant similarities. Yeah. So let's check out some of these distal hybrids. We've got a magnificent rifle bird. It's a very beautiful bird. It opens up its wings in a beautiful display, and it makes a sound like a rifle shot, which is where it gets its name from. And the 12-wired bird of paradise, quite extraordinary, with these bright yellow feathers. And you can see the way the shaft of the feather extends out towards the base. There are 12 of these shafts. And for a long time, it wasn't understood what on earth they were for. And it turns out that when he's hopping up and down on a sort of a naked branch or whatever, trying to call out to his sweetheart, if she comes along, he will tickle her on the beak with those shafts, those little, yeah, and apparently she really likes that because gets dreadfully interested. If these two very, very different birds, you cross them, you get Manitou's rifle bird. Now you notice that the greens have gone, the yellows have gone, those 12 wires have gone. It's kind of gone back to this sort of compact bird. Let's check out another pair, shall we? Here's the Western Parotia. It has these, it's the males that usually do the fancy display. Can you see these feathers at the back? When it dances, it whizzes these feathers around its head and it fluffs out its feathers. It looks like a ballerina. It's quite extraordinary. And it clears out an area on the forest floor, leaves it naked of leaves or anything, so it's staged to dance on. It's most extraordinary. We're going to cross that with the long-tailed Paradigana, quite a different genus. And you'll notice that there's this sort of beak, I'm quite so sure what you call it, protection or something, that yellow sort of mask. And when you cross the two, you get the same kind of species again, this hybrid, which sort of loses a lot of these spectacular things and goes back to being sort of a rather dull, almost, at least compared to its parent's bird. But obviously the information from both sets of parents are present. You could just select it all back out again, it's all there. Yeah. There's another picture, the ones highlighted in blue, they're the distal crosses, and you see they're going back to this original species. The information was always present. Good, how are we doing for time? Do you have a little bit of time left or not?
Still having 5 minutes, yeah.
5 minutes, let's see what we get with this. All right, so once again, something quite different, just to show you what's going on and to try and understand then principles here. So this is, I'm sure many of you have seen cabbage white butterflies. They're very common. They look like white butterflies. If you ever actually stop to catch them, you see little black dots on them. Yeah. They arrived in New Guinea, and something startling happened because the base of their wings suddenly decided to get really, really fancy and painted up. Wasn't there before. Suddenly it starts in New Guinea, and it starts in the information I have is from the left side of New Guinea. The whole of New Guinea looks like a bird of paradise. So right at the top, that's why it's called the Bird's Head Peninsula. And there's these mountain ranges, and this is where you find these butterflies. I don't have enough time to go into details now, but just briefly, these are, this is one of the species groups. I forget the number, it's 118 different species, divided into 17 species groups. So this is one of these species groups. It actually has 11 species, and I'm only showing you 6 of them. And these patterns that they show are absolutely characteristic of those specific species. They're fixed in. And what is interesting is these butterflies are always vigorous and healthy. Quite often you will see repetitions of some of these shapes between the different species, and you'll see they're consistently fit. Here's another species group. These butterflies are gorgeous when you see photographs of them. And another species group, well, I could show you more. The dorsal side is boring. It's just black and white, like a cabbage white. Here's the topper one. But underneath was all of that spectacular change. The observations drawn from the vigor, because they're all vital. It's not as though some of these, if they arose by mutations, you might expect at least some of them to be sort of on death's door or whatever. Mutations aren't a good thing, right? But they're always vigorous. And you find patterns recurring as though the information's already there. And the other thing you find is that there's a consistency about their fitness. Why should we care about that? Well, you remember that if these things arise via mutation, we're always trying to select for something which is fitter. So we should see some kind of fitter butterflies, but they're always equally fit. They're consistently fit. Yeah. All of this suggests, that's what these 3 things are always seen. Yeah? Numerous adaptive radiations found in it: silver swords, honey creepers, cichlid fish, dewlap lizards, etc., etc. Yeah, the conclusions drawn are always the same. Where abundant diversity observed, it results from meiosis and hybridization, recombining pre-existing genetic programs. It's a logical consequence of Mendel's findings. How quickly does it occur? Well, because I'm short on time, I won't say too much about that, but in a situation where there is self-fertilization, it can occur very quickly. So within about 20 generations. Here's the geckonic lizard adaptive radiation. They all belong to the same lizard group. Lots of fancy pictures. This is an adaptive radiation of dinosaurs, which requires tens, maybe scores of generations, but it doesn't require 150 million years. Mendel observed that at each generation, heterozygosity was lost, and it was lost in 50% of the progeny, and the other 50% suddenly were homozygous. Now, as soon as they're homozygous, there was what is referred to as being fixed. It could be fixed dominant or fixed recessive. And it's the different combinations of these dominant and recessive which gives you then all of your different varieties. He worked out the math of this. It would take far too long just to go through this, but these were just talking about the math. What I do want to do is to take us to this picture where we look at loss of heterozygosity in populations. And it was a study, a very famous study done by a guy called Bory. And what he showed was that if you start with heterozygous flies, that within a short time, they will either be all homozygous dominant or all homozygous recessive. It just happens. Heterozygosity is lost. I was going to tell you about fixation, but I won't. But I want to show you this picture because I'm short of time. I'd just like to show you this and then jump to the conclusions. But self-fertilization, it was one unusual factor in Mendel's experiments, because usually we have bisexual populations, but in plants, obviously the male and the female parts are both together in the same flower, and it just so happens that peas normally self-fertilize. Under those conditions, you get these species arising very quickly, within 20 generations at most. So what happens if they are sexual populations? What happens there is a process of genetic drift comes in. We can talk about this later if you want to, but it all becomes very mathematical, and really you should speak to biostatisticians like Ola about that kind of thing, but we can discuss that later. But this shows you what happens in a panmictic, which is a fancy way of saying a sexual population, of different sizes. Because if you look at the blue line, which quite quickly drops to 0, that's how quickly these new species would arise. But that's for small populations. However, large populations take longer. And so what you find is this is a mechanism which is clearly going to work on islands or in lakes or something like that. You're going to quickly get some new species. But where there's a big population established, you can maintain the potential for diversity. You will always find the most pan-dominant type as the main type there, but you do maintain that variability for many generations. Good. Having said all of that, it's easy how we can get all of these different trogons. It's easy how we can get all of these birds. This is the way we do it. Mendel did ask one question. He said, does this allow us to have unlimited genetic change? The answer to that is definitely no. When the molecular genomic studies came out, it was shown, this is the way we get our different species of gibbon. That's just a genus. Yeah. It's also the way we get these cichlid fish species. Now that they've actually been studied, it's been shown that this has caused hybridization between two divergent lineages, facilitated this process by providing genetic variation that subsequently became recombined and sorted into many new species. This is what the science is telling us. How does a whole family arise? Remember our cats? We know they can cross with each other. Here are some Liligers and some Litigons. We got the Leopons here and the Jagleons. But what has rather recently came out, I think it came out last year. No, it's a little longer than that. But what they found was, when they were looking at this, they found that there was recombination going on, not just that could be seen, but there was evidence for it in the genomes that they sequenced. If you're interested in any of these papers, I can share them with you. This process can give rise to whole families. These are our baramin. And this explains where speciation comes from in baramin. Species and families normally arise via Mendelian speciation. The genetic information required to form even the vast number of species that arise in exceptional adaptive radiation is already present in the genomes of heterozygous ancestral species Although mutation occurs, none is required to produce the plethora of species observed. Observations in the lab and in the field demonstrate mutations are almost always detrimental. It's not mutations. These things arise, and Mendel told us how, and some time ago already. Thank you for your attention.
So thanks a lot, Nigel, for this fascinating talk on Mendelian speciation.
I think. Is it this?
Yeah, thank you.
Does it work now?
So, does it work? Maybe I can just speak loudly.
Stand close together.
We'll stand close together, yes. Oh, try that one.
Hello? Hello, can you hear me?
If you stand right a bit closer to...
Okay, so yes, so thanks a lot, Nigel,
You're
for
very
this
welcome.
your fascinating talk on Mendelian speciation, and it's an eye opener that Gregor Mendel in the 1860s formulated this exponential law of speciation, and he was really prophetic, because this is turning up now in recent research.
It was also why he was doing those experiments.
Yeah.
He was actually providing, because he knew about Darwin's book, the German translation had just come out. He had a copy in his library, and he was explained where this comes from. Because Darwin had no idea. I mean, how could he? He didn't know about DNA or anything like this. He'd not done those experiments which Mendel had done. He went off on a complete tangent and came up with an idea which is quite incorrect. That's not meant to reflect badly on him. He was actually a fine biologist. Unfortunately, he didn't include his faith in what he was doing, which is a shame, but otherwise... so Mendel was answering those questions. He was explaining. He even said what he was doing and why he was doing it, and that it was to show where speciation came from. And yet people, I don't know if they just don't bother to read his paper. There are fine English translations, and it's definitely worth reading. It's fun, it's very rich, the language which is used.
That's fascinating.
Yeah.
So now we have time for questions. So.
Many species are rather constant in nature, that have changed so much. Is the reason you think that they are largely homozygous?
That would be an immediate...
Or that the populations are large?
Oh, yes, right. So the question was, why are some populations rather homozygous? Yeah, or they stay the same over a long time. And there will be two answers to that. The first would be, there is this tendency for things to move towards homozygosity. And of course, as soon as a population becomes homozygous, unless it finds a hybrid, it's going to stay that way. Yeah. And with these large populations, it does seem as though there can be some variation sort of remaining, but it's mostly hidden. Yeah, it doesn't come out in the same way. So first of all, I would say that it's this gain in homozygosity which keeps them the same. It does make a difference. On Hawaii, when the honey creepers came, we have a little dicky bird in Michigan. It's called the rose finch. It looks like a little, sort of like a red-breasted and crested, well not crested, but red-headed sparrow. This is the rose finch. And it looks as though when they went to Hawaii, whatever it was, transposons under new environmental conditions, got triggered. And then these beautiful honey creepers, all those different colours and fancy built, came out from that. That's what the molecular evidence is pointing us towards. Yeah.
So, Frank?
I think it was before here. But
is there these hybrids? Have you ever seen any kind of direct head-to-head comparison on the sequence level, on the DNA level, on the sequence level, directly to see
that information
to reflect the same thing you?
Okay.
When you say everything is still there.
Yep.
Can Could you have shown that on the DNA sequence in itself?
So I've been asked about, has this, what has been described, or this speciation, has it been shown in species? It was actually why I wanted to show you these last three pictures. So this is Papillot. This paper came out last year in Science. Yeah, and there the genome sequencing was done. And then it was formally proven that this was the process going on. So if you're interested, it's from a Sorensen, so perhaps someone from nearby here.
You can see half-half, kind of half-half DNA from both sides, kind of saying in the sequence, and really observe.
Yeah, they sequenced the species and could show where that came from.
Yeah.
So that is formally proven in this for this genus.
In this paper.
Yeah. And then in this one, this is they call them a species flock, yeah. And we're talking now about there are a total of 4,700 species. But when they look for that, this is from 2016 from Maya. They show that this was also due to hybridization. So they sequenced the genome. There's another paper on the same thing, also related to these flocks, but with fewer species, which show once again exactly the same thing. This is proven now. This is standard biology, and as I say, you're very welcome. I can pass on these. The other one was for the cats. I can pass this information on to you. You can read the original papers, but there was this genome sequenced for these different species, and that's why they know that. They do crazy things now. They've sequenced all of the different species of butterflies in Europe, and butterflies in hundreds and hundreds of species have the genomes for the sequences. It's astonishing work. Questions?
Okay, we can.
I think of the period before Noah.
Yes.
Can it be said something about, would you agree that there will be very many of those different types already were there before Noah?
I could well imagine that as far as land animals go, that there would have been other land animals before Noah that have either gone extinct since or as a result of that flood. But that's wild speculation on my part. Perhaps there's fossil evidence for that. Yeah. What is interesting is the whole question of when God created the animals, did he, like he did with man, just create a single pair? Or were there quite a few? Because if there were quite a few species or not, individuals of a species created, there could have been a great deal more genetic potential already available. But obviously then, at the time of Noah, after the flood, there was this bottleneck, this important bottleneck. Yeah.
We have a question here that wasn't...
Okay, yes, sorry. Yeah, Nathaniel, yeah.
So would you say it's the rule or the exception that hybrid species are sterile?
Yeah, so the question was, are hybrids, do they tend to be sterile or not? There was this sort of cultural meme that they were always sterile. Based on what I have looked at, about 50% of the time they are, and about 50% of the time they aren't. There's lots and lots of hybrids that are definitely not sterile. Yeah. What did I want to say about that? There was another point I was going to make. I can't remember now, but it was, yes. So that would be my answer to that. I don't know why there was some... I think it was because such famous biologists said no. You have to have reproductive isolation between species in order to keep them separate. If they can rehybridize, all the genes can come back together again and you'll start to lose your species. And there are a couple of species. A very famous example is the red wolf in the United States, which has basically lost its identity now because there's so few of them. They'd started to mate then with dogs, just domestic dogs or whatever, or wild wolves, and then we basically lost them now. But does that start to answer your question?
So, last question, Peter.
Well, it's not a question, but I think if I look at this presentation, and I also think about my own presentation, then we have in fact a full-blown creation science theory that explains what we see in nature.
Mm-hmm.
So I think what we should do, we should write it down.
Can you stand close? It's great if we get your comment, I think.
Okay.
in the mic.
Okay, is there a mic? Oh.
Yeah, if you just stand close by, it will be good enough.
What I just made as a sort of a remark or a summary, if I look at what you presented, and if I think about what I presented myself, we have, I think, a full-blown theory of how all these species came about in a creationist framework, a creation science framework, I would say.
So I'm very much looking forward to Tony's talk tomorrow, Tony. Yeah, but he'll be talking about his epigenetics. Yeah. Yes.
They complement each other.
Mm-hmm.
Yes.
It's a great note to end on, I think.
Yes.
Yeah, I think so. It's really excellent comment because we see that we look at different mechanisms of speciation, and now we have heard great talks about different ways of creating the bushes in the bushes of light models. So,
and also in a relatively short time period.
yeah, yes, they're very short. You showed that with your hybridization experiments. Yeah, very impressive. So, let's give Nigel a hand.